Welcome to this session of Shadow the Scientist. My name is Jamaica Marshall, and I'm the Shadow the Scientist Programs Manager. Today, our guest scientists are Michaela Capage and Jessica Bonner of the Massachusetts Institute of Technology. Now, the Shadow the Scientist initiative is brought to you by an incredible team at the University of California at Santa Cruz. We are funded through a generous grant from the Heising Simons Foundation. Now, in this session, we will be exploring the process of imaging Drosophila larval ovaries with Michaela and Jessica. Now, they are already ready to begin. They are live in the lab, which you can already see behind them. So, without further ado, I'd like to welcome Michaela and Jessica.
All right. Hello. Jess and I are both very excited to be here. Jamika, will you allow sharing so that we can share our quick presentation before we move to the microscope?
Absolutely, that should be there. Oh, there you go.
Got it.
We're
Thank
in.
you.
You're welcome.
Okay. So hi everyone. Today we are going to be, as Jamika said, imaging Drosophila larval ovaries. So for a little bit of background, before we get in, the model that I use in my research in the lab is the developing Drosophila melanogaster ovary. So Drosophila melanogaster are just like your basic fruit fly. And they are a really fantastic model for developmental biology and for basic research in general. And particularly, we in my lab focus on the what's called germ cell biology. So how the fruit fly makes eggs that then allow the generations of flies to continue. And the cells that give rise to eggs are very important because without them, the species cannot continue. However, they are completely dispensable for the health of the organism. So the female fly is completely fine with non-functional ovaries. However, the species wouldn't continue without that. So an organism puts an immense amount of effort and control over how ovaries develop. So on the slide here, I have just a sort of schematic of what the adult ovary looks like. So the ovaries, each female fly has 2 ovaries that reside in the abdomen. And then the ovary is actually a repeating pattern of these sort of specialized egg like conveyor belts, basically. So the cells at the very, very, very top of the ovary are stem cells, which will asymmetrically divide to produce egg precursor cells. And we call these germline stem cells. So these germline stem cells will reside in the adult ovary. However, they will actually develop in the larval ovary. And at this point, we call these cells primordial germ cells, or PGCs. And we call them primordial germ cells just to sort of denote that they are not fully developed yet. Like they are in the process of becoming germline stem cells, but they're still in this intermediary developmental stage, and that's where I'm studying them. So they develop in the fly's larval ovary, which is on the screen here. The goal of the experiment that I'm conducting today is to count the PGCs. However, if you just look at this image, which is marking all of the nuclei inside the ovary, you can't tell very much about what's going on. Like, where are the PGCs in this image, or how many of them are there? I don't know just by looking at the DNA of the cells. And so I have to do an additional protocol before imaging called an immunofluorescence protocol. And this identifies PGCs. So on one side here, we have what the ovary looks like before I do the immunofluorescence, where it's just the DNA is marked in gray. However, after doing the immunofluorescence protocol, I can actually identify the PGCs based on their expression of a particular protein, which is marked in magenta. So basically all of the cells that we see that are pink are these PGCs. And what the protein that's actually being marked is a protein called VASA, which is a very sort of central protein for setting PGC cell identity. And so if I zoom in a little bit on what this image looks like, basically the cells that have VASA, this magenta color in the cytoplasm, those would be identified as PGCs. And those are the cells that I'm interested in counting. In addition to VASA protein for the PGCs, the images that we're going to see on the screen are also going to have traffic jam protein in the nuclei of other cells. So let me back up for a second. Basically, the fly ovary is comprised of PGCs and non-PGC cells that are important for supporting germline biology throughout the whole adult life of the fly. And so we're seeing two different types of cells in the ovary, ones that have PGC, the VASA throughout the cell, those are the PGCs, and then other cell types are going to be marked by two different colors. They're going to have a protein called traffic jam, which is going to be located in their nucleus. And then they're going to have a protein called the sodium potassium ATPase alpha, which is a very long name, but it's basically one of two subunits of a protein called the sodium potassium ATPase. And that protein exists on the membrane of the cell. And so the images that we're going to see, we're going to see some cells that are magenta, some cells that have red on the membrane and green in the nucleus. So the final product image that we'll be seeing on the screen today is going to look something like this. Lots of cells with red membrane. Some of the cells are going to have green in the nucleus, and then the cells that I'm most interested in, these ones with VASA magenta in the PGCs. So a couple of final details. During my imaging session today, I'm going to be taking a type of image called a Z-stack. Basically, this is a high resolution image throughout the entire adult larval ovary. So starting at the very bottom of the ovary and working all the way through the entire extent of the organ, I take a slice every quarter of a micron. So with these automatic images, I can employ an image analysis tool that will automatically count the PGCs based on their VASA expression. And so what I'm showing you in these two videos is a Z-stack throughout an entire ovary, showing all of the PGCs that are going to be appearing and disappearing as we move through the organ. And then in the other video is kind of the version of the PGCs that this computer program sees. And what is really nice about this is that this program will automatically identify these cells and count them so that I don't have to do any of that analysis work manually. So our first video here and our second. And what I hope you can see, the video is running a little bit fast, but basically as a cell appears on one side, a mask of the cell appears in the other video. And these masks really accurately capture which cells are present and where. And then the program is sort of keeping track of every single mask that it calls and then spits out this quantitative thing at the end. So with that, we are going to turn to the microscope and start imaging some samples together. So we're going to stop sharing.
We do have a question actually about that process, if you don't mind.
Yeah.
All right. This is from Raja Guhathakurta, of course, the Shadow of the Scientist founder. So in the immunofluorescence image, is it significant that the magenta color marking the VASA protein appears to be concentrated in the outer parts of the cell?
Yeah, so that's a great question. So different proteins that reside in cells do have sort of specific domains of the cell where they're localized. So I think I said before, there are some proteins that we're going to be looking at that are on the membrane, some in the nucleus, and exactly VASA is located in the cytoplasm. The fact that VASA is in the cytoplasm is important for PGC identity. Based on what the protein does, it has to operate in the cytoplasm of the PGCs. For my imaging and for the sort of process of analysis, the computer program that my collaborator and I developed knows to see and knows to expect that sort of donut shape of VASA pattern. And so when it sees that sort of donut shape with a hole in the center and the signal in the sort of cytoplasm of the cell, it recognizes that as a PGC. However, we trained that program specifically to see and expect that pattern. We could have trained it on something in the nucleus or potentially something on the membrane. But basically what is important is that it is very consistent, because the program is expecting to see these donuts. If it doesn't see them for some reason, it doesn't know what's going on. Yeah.
Thanks, Michaela. It sounds like or it looks like the nucleus of these cells is quite large compared to the cell size. I mean, you could have cells where the nucleus is tiny. That accentuates the sort of ring-like feature, right? If the nucleus were tiny, it would mostly be filled in, with the cytoplasm would occupy most of the body. But here, it doesn't seem to be the case.
Yeah, that's a great observation. The nuclei, so the germ cells in general, are very big compared to some of the other cells in the ovary, and sort of generally compared to other cells that exist in the Drosophila. And they also have, you're totally right as well, they also have very big nuclei. And I think that that shape and size is actually very consistent across all of the samples. It's sort of a very consistent feature of what these cells look like. And so I think that's the other benefit for why this automatic program works so well, is because there's a lot of consistency in what the samples look like, week to week and even year to year in my experiments. And so the program is very much expecting something that is that shape, generally in those dimensions. But I think that the size and the relative size of that hole in the nucleus is definitely part of what the program is understanding.
I'll say one more thing, and I promise I'll keep quiet after that. This reminded me about analysis we do in astronomy, where you have a bunch of stars, and they're close enough together that blending is an issue, that the program will sometimes put a circle around something that's really two stars. The brain can sometimes tell it's two stars, but the program has put one mask around them, or vice versa, where it misses something because it's too faint or small compared to something else. I bet you have this issue as well. It's not 100%.
I could go into this if you want a little bit.
Yeah, so it's definitely it's not 100%. However, what my collaborator has done is actually fine-tuned this model over many iterations. Certainly when we started working together about 2 years ago, the very sort of first version of the model was doing a lot of stuff. It was clumping a lot of stuff, it was splitting a lot of stuff, it was all over the place. And so what my collaborator did is he created something called a ground truth data set, where he actually hand counted every single cell. And so now he has a data set where he is sure how many cells are in that sample. And then he can run different models against that same data set and basically see how well the models are doing compared to that ground truth. And so we, I think, are on our third version of the model. And it is now hitting about 99% accuracy to that ground truth data set.
Oh, thank you. This
Yeah.
is a great citizen science project, asking people to count these cells, right? Would be an amazing citizen science project.
Yeah, absolutely. So now I'm going to turn us to see the microscope. And this machine is quite large, as you can see. It's in this sort of specialized room that we call our scope room. And basically you've got a lot of machinery, and down inside on the stage, can I grab your computer?
Yeah.
I do think I'm gonna come in. down on the stage there, surrounded by that yellow tape, is the slide where the samples are sitting. And I hope you can appreciate, like, the machine that we're working with is huge. The sample, the slide is very small, and the samples are very small. So we're putting quite a lot of power on something that is, like, very tiny, and yeah, not very big compared to the sort of machine that we're running to look at them. So I operate this microscope almost entirely through the computer behind me. So what we're going to do now is switch over to screen sharing from the computer. So what you'll then be seeing is the sort of image software that I use to acquire the images. So let me switch over to that.
We're also going to turn off the lights, and so Michaela and I are going to become shadow people, but we're still here and very happy to answer questions. This is actually pretty essential for Michaela's experiment so that she can image properly and the ambient light from the room doesn't interfere with the excitation and emission of the laser.
So actually, before I turn to the machine, I want to make sure that there's no questions in the chat, or if anyone has anything that they want to add.
No questions currently.
Awesome, great. And Jess and Jamika, I assume you'll let me know whenever there's something that comes up.
Will do.
Okay, cool. So the very first thing that I am doing here is before our imaging session started, I located all of the Drosophila ovaries that I had put on the slide. And so what I'm going to do now is I'm going to navigate to one of my saved positions.
Would you screen share?
Ooh, definitely.
Thank you, Jessica. I was just about to ask that.
No problem. Michaela is multitasking. Also, by the way, I am Jess. I am not a lab mate of Michaela's, but I am a peer and a friend also in biology, but in a very different subfield of biology. And I'm here to answer questions when she is concentrating.
And it is so helpful in these sessions to have somebody emceeing it a little bit, because sometimes I, will get very, very focused. And so, yeah, Jess knows a lot about science generally, but we're also very much here for sort of questions about the process of being a scientist, or coming from undergrad and starting graduate school. Like, we're, I think, very open to those questions as well. Okay, so now that I'm actually screen sharing, I can actually show you what I was doing before. So if you look over here on the side where my cursor is, we are looking at a list of all of the positions where the ovaries are located. So I'm going to navigate to a new position. The next thing that I'm going to do is navigate to the channel that is called the DAPI channel, and that is basically marking the nuclei of all of the cells that we're going to be seeing. And now I'm going to, it's called going live, but basically we're going to turn on the laser and start to actually see the image. So the very first thing that I do to start one of these experiments is I am going to set that Z-stack. So basically with a small dial on the side of the microscope, I am finding the very bottom of the ovary, which I can start to see is coming into view here with these cells. And actually, let me stop really quick. So I typically like to have my DAPI always in gray for people to see. That's what is on my images on the slides. So I'm just going to change what that looks like on the scope. So we go live. I'm at the bottom of, okay, well, it's back to blue. It's same thing, just blue. And now I'm sort of scrolling through the entire extent of the ovary, all the way through to the top. And then once the image starts to disappear out the other side, I will set this as the last position of the Z-stack. The next thing that I need to do is actually center the image in the middle of the screen. So we're a little off center right now. So I use a joystick also on the microscope. I'm going to scoot us into place here. All right. The signal that I'm acquiring here looks good. I'm not saturating any of the pixels, basically seeing too much signal from them. The image is like entirely within the extent of the screen. Now what I'm going to do is I'm going to check all of the other channels that I'm going to acquire. So next, I'm checking a laser that is exciting or emitting at about 488 nanometers. So this is the traffic jam protein, which is in the nuclei of some of the cells. Next, we have our membrane protein, the sodium potassium ATPase alpha here. And next, and finally, we're going to look at our VASA signal. So as I just scroll very rapidly through the entire extent of the ovary, you can see these VASA positive cells sort of coming in and out. The signal here is really nice. It's very low where there isn't a cell, and then it's much higher where there is actually a cell. And so that's very good. So I'm gonna stop. And now I'm going to hit start experiment. And what this is going to do is it is going to start from the very bottom of that Z-stack in DAPI, and it's going to acquire an image all the way through the entire ovary. Then it's going to go to the bottom and do the same thing with traffic jam, the same thing with this membrane protein, and finally the same thing with VASA. And so this is a pretty long acquisition. It's about 13 minutes, which I can see down at the very bottom under experiment information. And so once I get this started, this is now a great time. This is why these acquisitions are great for these Shadow the Scientist sessions, because we can just chat, answer questions as we watch the images appear on the screen.
So as we're waiting for this acquisition, and I love this, as Raja's already pointed out, to some comparisons to astronomical imaging, and this, of course, is another one waiting for the image to actually be fully acquired, so you have a bit of downtime. So could you talk about the process of learning how to use such a complex piece of machinery?
That's such a good question. So I think that it is, a very long process to be able to use this piece of machinery and use it well. And this is also really important because this machine is shared by everyone in our lab, and it gets an immense amount of use. It's booked basically 9 to 5 every single day of the week, including most weekends. And so it's really important when you're using this machine that you are using it correctly, that you don't damage it, that you don't jeopardize anybody else's experiments. And it's also very expensive. And so I care a lot about making sure that, I knew what I was doing before I started just using it myself.
Speaking as Michaela's friend, I will also say, from my understanding, her experiments really push this machine to its limits. The analysis that she's doing is really pushing the machine to its limits. As part of her experimental workflow, these are sort of the bread and butter experiments that she's doing. So to ensure she has enough instrument time, I'm quite certain she's in at least one day a weekend, if not both days, because it is a shared instrument and she doesn't want to consistently not get in the way, but occupy the instrument when it's shared by, how big is your lab? 20 people?
15. There's about 15 of us.
Yeah.
Yeah, that at some point are using this scope. Yeah.
So when you have such a valuable resource, and I think this is probably actually true for your astronomical telescopes as well, there's a huge amount of effort put into organizing everyone's time around being able to use such a powerful instrument.
Definitely, yeah. And so in terms of training to use the machine, the way that my lab runs our trainings is that the first thing you do is someone operates the microscope and you sort of sit in the corner and you watch and they sort of explain what they're doing. And you do that for about 2 hours while you acquire some images. And then in the next session, you are the one clicking the buttons. After the person who's training you, it tells you what buttons to click so that you don't click anything wrong. And you do that for about 2 hours. And then in the third session, the person who's training you is the one who sits in the corner and is mostly silent while you click buttons and look to them to make sure you're not going to click a horrible button that's going to break everything. And so I did about 6 hours of training with the machine. And at that point, I was considered fully trained and able to use it myself. But I still ask lots of questions about, when things are a little bit weird with the microscope from some of the more senior people in the lab.
And Michaela, you mentioned this earlier, but you said that these Z slices are very, very thin. So for the larval fly ovary, it is incredibly small. What is like the distance range that you're imaging right now in terms of your
From
first
top
Z
to
stack
bottom,
to your
yeah.
last Z stack?
So this one is they're all about 50 microns from top to bottom. So that is 50 times 10 to the negative 6 meters.
Yep.
And I don't know that that's a helpful number.
I will convert it to a helpful number. But it does bring up the point that these fly organs that you're working with are really quite small.
They are.
And you're like one of 5 people in the world who can do this, right?
So basically this technique is called a larval ovary dissection, and there definitely are not that many labs that do this. Partially because there's not a massive number of people who study Drosophila ovaries in general, but the larval stage is considered very tricky to work with as well. And so, I've actually gotten a lot of emails from other labs, and I've actually done some sort of like consulting experiments where people will send me their flies and I will actually dissect and image it for them and then send them the images. I've done that once, just because there's not that many people, who do this.
And so to put the number that Michaela said into a more maybe like thing we can understand,
meaningful.
it is half of 1/1000th of a centimeter is the distance that Michaela is imaging from the first image she took to the last image she took. And you're taking how many images?
So then I image every Z slice is a quarter of a micron apart from one another.
Yeah.
And so on average, it's about 200 slices per sample. This one actually, if I take you back to the scope, this one is 229 slices.
So just to give you an idea of how the incredibly fine adjustments that this microscope is making, that's equivalent. Every image that you're seeing compared to the last image is 1/100,000th of a centimeter. So that's part of the reason that this microscope is so expensive.
It has
the ability to move 1/100,000th of a centimeter between images.
Yeah. And the reason why the Z stacks have to be so close together and so many of them is actually, Raja, exactly what you were saying with the model sort of clumping things together or like separating things apart when it's actually one. Basically, it's called Z resolution. Basically, how finely resolved is the image in the Z direction. And the very, very, very thin slices allow the Z resolution to be very high, which means that the program has a lot of information as it tries to decide like where is one cell stopping and the other is starting and whatnot. And so so basically as I worked with my collaborator in developing this protocol, we started with differently sized slices that were a little bit bigger, and then like that wasn't working for the program, so my collaborator said, okay, can we make them smaller? So it was very much like a back and forth between us about the analysis side of things and then actually the image acquisition side of things as well.
For absolutely, Jamaica. I was going to say if any of the students in the chat have any questions whatsoever, it can be very specifically about the experiment that Michaela is doing now or more broadly about science. You're welcome to unmute or post them in the chat, whatever you're more comfortable with.
That's right. So I have a question about the lab. Is this the Ruth Lehman lab that you're in, Michaela?
Yeah. So my advisor's name is Ruth Lehman, and she is a faculty member at MIT. And she is a very well-known Drosophila biologist who's been studying germ cell biology in Drosophila for a very long time.
Okay, I thought so. I wanted to check to be sure. So Manisha, and this question's for Imraz, Rhea, Barty as well. I'm curious, what opportunities, if any, the students have had to get into a lab and use this kind of equipment or similar equipment, or even be a part of research project.
Yeah, I would first ask the students if they would like to add from their experience. Emrose, Ria, anyone, if you would like to add, or Bharti?
Hi, I'm Ria. I'm an undergraduate student from Ashoka in India. Um, I am currently doing a thesis where I'm working on the Drosophila ovary model itself. Um,
Oh my
What are the chances?
yeah, I'm looking at the impact of dietary modulation on the ovarian stem cell behavior in Drosophila, and
Yeah.
which is why I thought this would be interesting to join. I do have like more than a comment, like I'm just amazed by how you're doing larval dissections because ovary dissections in general, like I keep losing my mind. I'm also very new to this. I've been doing this for I think almost a year now, but not too long. But yeah, I mean, how did you like practice that?
Well, it's so fantastic. It's so nice to meet you, Raya, and to like have someone on the Zoom who, yeah, knows these flies kind of personally. Um,
Yeah.
the you just said a little bit about your research, but the role of like diet in the and nutrition in the germ stem cell, those are always super interesting papers to read. I
Yeah.
really like those, so that's really cool. The in terms of learning the larval dissections, so when I joined the lab, there actually was no one who did the protocol
Mm-hmm.
currently in the lab, but I did have someone who had previously worked in the lab and then left for another position, recorded videos of himself doing the dissections, and those were sort of like saved internally in the lab. And so I was able to watch those videos and basically try and replicate what he was doing.
Mm-hmm.
But it took a really long time. It was like several months of just kind of showing up to lab every day and just destroying a bunch of ovaries and larvae and just getting nothing, and then being like, okay, well, time to go home, and then
Fair
I'll
enough.
do this again tomorrow.
Yeah.
And it was a very long process, and it was also, I mean, I'll be very honest, there was like a lot of doubt. I think, like, is this the right approach to a PhD? Like, I'm not getting any data, I'm just sitting here destroying ovaries.
Yeah.
It was a very like long investment, I think. But it, more than anything, is just practice.
And I think
Yeah. Yeah.
something that I don't know, well, you have just demonstrably experienced in your PhD is sort of doing something that people around you aren't doing. I have also experienced that in my lab environment where you're bringing a new technique in that folks aren't familiar with, and I'm sure this also happens in astronomy as well. But I think one of the challenges of doing the science that you find the most exciting is oftentimes it requires you to learn independently and innovate and apply things a little bit in a vacuum where you're sort of making mistakes and not always doing the right thing. That's a very common trend in grad school, I would say.
I should
There's no
keep that in
sample
mind.
destruction in astronomy. There's no destruction of stars or galaxies that
That's
I
true.
can
Okay,
It's
there is
true.
no sample. Yeah, astronomy is a little higher stakes in that way. Sorry, Rayo, were you saying something?
No, no, I was just laughing at this.
Okay, that's true. There is no destruction of stars yet. No, I'm just kidding. We don't want to do that.
We don't want to do that.
Any other? Rhea, it's so cool to hear that there's another Drosophila
Yeah, I love it.
Yeah.
researcher. Are there any other comments or questions from students? Even if you are not a Drosophila ovary researcher. We would love to hear anything you have to say. Oh,
Okay, our image is done.
pause that. We'll come back.
Pause that. We'll be right back. Okay, so image has acquired. Looks good. Now what you have to do is you have to save your file so you don't lose it. This is the 5th ovary that I'm imaging today. All right. So now basically the process just repeats. Move to our newest position. First, set our Z-stack in DAPI.
And while Michaela's focusing, I actually just wanted to draw another parallel between astronomical telescopes and biological microscopes, which Raja mentioned with his initial question, this idea of resolution. And so Michaela's talked about how with this microscope, the way that she's resolved resolution is by taking finer, or the resolution issue of two light sources being conflated with each other, is she's taking finer and finer Z-stacks, which allows the software that she's trained to identify cells that are separate from each other. I work in microscopy that images things even smaller than cells. So I work in electron microscopy. And there's very cool microscopy techniques that allow us to get to even greater and greater levels of resolution. Something that's fluorescence microscopy with higher resolution. And you can't manipulate your astronomical sources in this way, but you can make these different dyes that you're using actually blink. So a dye will light up at one time, and a dye very close to that dye will light up in a separate time, and that will allow the light images to alternate back and forth in a way where they're not clumped together, because we're seeing them imaged at slightly different times. And this is something called super resolution microscopy, that in actually controlling the environment of your sample, you're able to get to higher and higher levels of resolution, because you're actually imaging things at slightly different times. Or not at slightly different times, but they're emitting light at slightly different frequencies. You have something to say?
Which is really cool.
Yeah, it is really cool. What do we got here?
Okay, so I wanted to basically I started this experiment earlier this morning, and when I was imaging it, one of the ovaries, I noticed that there was this very cool cell that is actually preparing to undergo division. And the reason that I know that is because many of the other cells have this like the blue signal here, which is the DAPI, is very diffuse throughout the nuclei of the cell. But if I zoom in on this one, what you can actually see is that the DNA has condensed into the shape of a chromosome, and Drosophila have 4 chromosomes, which you can see here, 1, 2, 3, and 4. And they are preparing to divide, I think. So basically they are, these chromosomes will line up in the center of the cell and then pull apart. But we're sort of, basically when I took the sample, I sort of like interrupted that process in the cell. And actually, I should say one thing. So Drosophila actually have 5 chromosomes. They have a sex chromosome, and then they have 4 autosomes. However, the 4th autosome is so small that there's almost no genes that sit on that 4th chromosome. And so it's possible it's sort of like behind, maybe it's over here, but typically you only really see like the biggest 4 chromosomes in a cell. And so I thought that was very cool, so I wanted to share it with you guys.
I did notice right before that gorgeous composite image came up showing that your Raja asked, will the division be meiosis or mitosis?
That's a great question. So these PGCs, these primordial germ cells, are sort of still in this progenitor state, hence their name, the PGCs. They are not yet germline stem cells. And so they are basically dividing symmetrically and they are undergoing mitosis. So they are just doing the same sort of normal divisions that all of the typical cells in your body or in the Drosophila do. They are not doing this sort of specialized reductive division that is meiosis. But that's a really fantastic question because obviously these cells are the precursors to the ones that will do that specialized reductive division.
I did note right before we got that composite image, there was a student who seemed like they were maybe unmuting for a question.
Ooh, and we'd love to hear it.
And then we turned to that composite image. So I don't know if that is still the case. And actually, Michaela, would you mind going? Oh,
Hi.
hi. Yes, excellent.
Yes. Hi. So Imro's here, and I also worked in a Drosophila lab,
Oh!
and yes. Um, and what I was wondering, because I work with infections, I basically the project I was working on, we had like two Drosophila populations. One evolved on like sort of a heat adapted line, and one was on ambient temperature. And what I was looking at that how infections sort of affect differently in these two populations. Now, after looking at this sort of ovary dissection, I was wondering how would a ovary of a infected sort of Drosophila would look like, especially if the larva is coming from an infected mother or the larva itself is directly infected.
That's really interesting.
Like what
Yeah.
kind of sort of changes we would see there?
Yeah, that's an interesting question. So my first guess or answer is that I think it would depend on what kind of infection and sort of what part of the Drosophila is affected or sort of what part of the fly's normal physiology is being impacted by the infection. So that's, I think, like the first thing is that it could have like very variable results, kind of depending on the details. However, one guess that I would make overall as well is that generally, I think that poor nutrient conditions or sort of poor environmental conditions, potentially infection, can sort of drag down the fertility of the fly. Basically, it takes an immense amount of energy to make all of the eggs that a female Drosophila will lay. And we know that certain stress, like heat stress or sort of various other perturbations to the living conditions of the fly, dietary stress or nutrient stress, will dampen how many eggs the fly will lay. And so it's possible as well that an infection that sort of drags down the health of the fly would mean that there could be issues with creating these progenitor cells or with the health overall of the germline stem cells in the adult ovary, basically impacting how fertile that fly is.
It's a great question.
Yeah.
Thank you.
Yeah.
Thank
I'm
you.
curious. I'm curious for the students who are attending, if any of you are considering research careers, or if you're considering careers that are not research as well, and sort of how you think about the role that research might play in your life.
Yeah.
Yes, feel free to unmute yourself to respond to that, Bharti, Rhea, or Imrose, or you can put your response in the chat.
And it is also totally fine if you don't plan to do research.
Yeah, we won't be offended just because it's how we spend all of our time.
Right. And while we're waiting for those responses, if they choose to share that, I did want to go back up to another question from Raja a bit earlier. The question was with the slices when you were talking about the thinness of the slices that you needed. Is this effectively tomography?
So I think I know the answer to that, but I'm actually going to turn it over to Jess.
I think tomography is a general technique that refers to concentrating an energy source at a certain distance along a sample point. So the reason Michaela points it to me is because there's also cryo-electron tomography, which is actually in the name of the technique, where you focus electrons at a certain distance within a sample so you can image it. But yes, tomography is a general term that refers to focusing the distance of excitation or tomography is a general term that refers to sort of taking an image at a certain point along a z-axis, and so yes, this is a form of tomography.
That's what
I guess what I should have asked is this effectively 3D imaging?
in essence, yes. Yeah. Because the Z resolution is so fine, I have very little loss of resolution in Z, which allows for almost a very good 3D reconstruction of the entire organ. Yeah.
And Z is distance into, we are looking at XY here, right?
Exactly. So what we're seeing here is XY, and the images are acquiring and pushing through Z. What I can actually do really quickly is show you, the program allows me to see orthogonal views. So if I change the view a little bit, so this box here is XY. Yeah, pushing through. Yeah, this box here is this is the XY view, and then these two views here are basically cutting it on the alternate axes. So I think this is pushing through Z, and so this is like ZY, and this is ZX. And I can basically change the Z position here, and you might be able to see this blue line is just basically running across the image. But if
Gotcha.
I instead change the Z position, or the Y position, sorry, we're now sort of running through the ovary on a different axis. And I think you can see this box here in the center is the cleanest, but the resolution, like you still can see what's going on in the other two boxes.
This is, astronomers use XY for the sky and Z for the line of sight, which is exactly what. Unlike
Exactly. Yes.
unlike
Yeah.
in biology, astronomers don't get Z information. It's a very important distinction. But there's another parallel. You've got this expensive piece of equipment that you talked about, and astronomers have expensive bits of equipment. And in general, you don't want to let human beings touch these pieces of equipment, right? So you have computer controls, which is so nice for Shadow the Scientist. You can share your screen. That's a parallel I see.
Yeah, I was, so I got connected through Shadow the Scientists through Jess primarily. And when I sort of heard about how it started with astronomy, I had definitely the same thought. I was like, I basically do the same thing. Like I'm using a computer to operate this very big machine, and I can share my screen and I can show you. And so I was really excited that, that there is this parallel, like some things are very big and far away and some things are very small and up close, but the way that we can share them with each other and with people around the world virtually is very similar. And so I really like that connection.
Yeah, and I do think a really interesting difference that Raja, you already brought up is and correct me if I'm wrong, but with astronomy, manipulating your samples is generally not something that you are all doing.
No, it's not.
Whereas biology, that is at the core of biology. When Michaela first set up her imaging today, the first thing that she always does with her imaging is image her control sample so that she understands the system that she has manipulated is behaving as she would expect. And what she's actually imaging right now is a sample where she's changed some variable that she then compares to her control sample. And for, I would say, the majority, if not, I'm sure there's a biological experiment where you don't manipulate your sample relative to some control. But for a lot of biological experiments, we're always comparing to a control sample. And I'm sure for the students who have done research who are in this session, you can absolutely relate that having those control samples working is incredibly important to the research that you're doing.
It's a great point, Jess. You can't reorient, you can't reconfigure in astronomy, but what you have is a very large N. So
Yep.
while you can't reorient a galaxy, you have so many galaxies that you can be sure you're seeing different ones at different, all possible orientations.
Yeah. Well, and actually, Raj,
That's right.
the way that you're talking about orientations, I was trying to think like what biology does not have control and variable samples, and actually, it's my biology. I do structural biology, and a lot of times the goal of structural biology is not to compare some variable to a baseline condition, but you're seeking to get an image of what the thing is in and of itself. So I am working to understand the structures of biomolecules that we don't yet have structures of. And as you said, Raja, the thing that you do with structural biology is I image hundreds of thousands of those molecules and I put them all together. And with that data processing, I sort of understand that in imaging many, many things, this end result that I get has some degree of truth to it. Michaela, you have something.
No,
No?
we've just set our third acquisition for the session. Yeah, but this is a great conversation.
Yeah, I'm curious actually, Michaela, like what is the variable that you're manipulating here?
So, so I am actually still imaging my control.
Later on the line.
Later down the line, later this afternoon, I will be imaging my my experimental sample. But what I am doing is, so I think I said this in the presentation, but the goal of my experiments is to count the number of these PGCs in the larval ovary. And so what I'm doing is I am changing the expression of certain genes in the surrounding cells. So if I go back to this image, basically what I'm doing is I'm changing the expression of a gene in the cells marked in green. So in these surrounding cells that we call somatic cells. And so I change the expression of a protein in those cells, and then I see how that alters the number of the magenta cells. So this is an example of something that is cell non-autonomous. So there's a change in one cell population that's actually influencing the biology of a neighboring cell population, which is a super interesting kind of initial starting place, basically, that these two cell types in the ovary have they really rely on each other for cues that guide their development. So the ovary tries to make sort of the right number of PGCs that's supported by the right number of somatic cells. And so my project is really about understanding how those two cell populations are communicating with each other and how they're informing each other's development.
I'm curious to know, since we have at least two students here in the session who are doing similar work, well, at least using the same kind of organism, what opportunities are there for potential collaboration, or even just to connect and have a conversation about research, if that's something the students would like to do?
Well, I would love to have more conversations with any of the students who are interested in research. I definitely will admit I can have more in-depth conversations about Drosophila research, but I'm definitely super available for sort of thinking generally about the process of research. But also, yeah, I think it would be really interesting to sort of hear a little bit more about the work that the students on the Zoom call are doing. I think that would be just fantastic. So in service of that, Jamika, please feel free to pass along my email.
Okay.
Yeah, and students can definitely reach out.
Wonderful. I will.
Likewise, but mine as well. But I think Rhea and Imraz, and I'm sorry if I didn't pronounce your names correctly, since we're both Drosophila researchers, I suspect that Michaela would be a fantastic resource for you both.
But I will share both of your email contact information, though. Thank you, Michaela. Thank you, Jessica.
But if either of you want to talk about structural biology or cell biology or biochemistry, I would love to.